My name is Jing-Ke Weng. Currently, I'm an associate professor of biology at MIT and a member of the Whitehead Institute. My lab studies the origin and evolution of specialized metabolism at enzyme pathway and systems levels. I'm honored to be a Beckman Young Investigator of Class 2016. Arnold Beckman is a scientist, engineer, inventor, entrepreneur, and a philanthropist. His life story has been a huge inspiration for me at the very beginning of my independent research career. Throughout his early academic research in chemistry, he saw tremendous opportunities in developing new instruments for chemical research. In 1934, he founded Beckman Instruments, which created many scientific devices such as the pH meter and oxygen meter. These innovations have revolutionized the research and understanding of chemistry and human biology. Following Dr. Beckman's spirit of scientific curiosity and entrepreneurship, I strive to push the boundary of science in my own research, while seeking opportunities to translate basic discoveries into real-world applications that will have positive impact on human health and the sustainability of planet Earth. My lab was supported by the Arnold and Abel Beckman Foundation to study the biochemistry of bioluminescence. Bioluminescence is the production of light by a chemical reaction in a biological context. In well-described cases, bioluminescence consists of the oxidation of a reduced small molecule called luciferin by an enzyme called luciferase with molecular oxygen. This oxidation typically produces a high-energy intermediate that decays with the production of an electronically excited molecule called oxyluciferin. Excited oxyluciferin then returns to its ground state, releasing a photon in a process analogous to fluorescent emission. The most well-studied bioluminescent systems are those of fireflies and jellyfishes. Fireflies use light as part of their mating behavior, wherein males display rhythmic flashes to attract females. The chemical identity of firefly luciferin and the gene encoding the firefly luciferase were discovered in 1960s and 1980s, respectively. Today, firefly luciferase is a widely used reporter gene in biomedical research. Bioluminescent cone jellies and true jellies are among the most prominent marine bioluminescent organisms. Study of the basic biology of bioluminescence in bioluminescent jellyfishes has led to the discovery of green fluorescent proteins. The luciferin of bioluminescent jellyfishes, calanthracene, is prevalent throughout the oceanic food web and is unique among luciferins in that calanthracene biosynthesis and calanthracene utilizing luciferases have evolved convergently in more than 20 genera. Although the general reaction mechanisms of luciferin-luciferase pairs are known for the major bioluminescent systems, the pathways that synthesize luciferins are not known for any eukaryotic systems. Supported by the Beckman Foundation, we set out to explore the unknown biochemistry involved in firefly and cone jelly luciferin biosynthesis. Using the long-read sequencing technology, we sequenced and de novo assembled the genome of fireflies Photinus pyralis and aquatic lateralis, native to North America and Asia respectively, as well as the distantly related bioluminescent clip beetle, Ignalator luminosus. Through a comparative analysis of these genomes, we discovered that firefly luciferases and clip beetle luciferases evolved independently. These high-quality genomes also provide rich genomic resources and a genetic blueprint for identifying genes involving luciferin biosynthesis. Using modern metabolomics and isotopic tracing techniques, we showed that firefly luciferin is not de novo biosynthesized in the adult or larval stage. We instead discovered a unique firefly luciferin recycling pathway, which is responsible for the maintenance of the majority of their luciferin. To study colanthracene biosynthesis in comb jelly, we generated a rich collection of tissue-specific RNA-seq datasets from Miniopsis lidii, a bioluminescent comb jelly native to the East Coast waters. From comparative expression analysis, we identified a set of candidate colanthracene biosynthetic genes, pinpointing to a unique metabolic system that recycles oxidized colanthracene back to colanthracene. We have also identified a major storage form of colanthracene in comb jelly, which was not known previously. My lab's other research has mainly focused on specialized metabolism in plants. Support from the Beckman-Young investigator program allowed my lab to leverage our genomics and the metabolomics expertise developed through studying plants to explore the essentially unexplored specialized metabolic systems in fireflies and bioluminescent comb jelly. This new line of work has yielded novel discoveries in an understudied scientific field. Moreover, these new findings will also enable future development of broadly applicable tools for biological and biomedical research. Being a Beckman-Young investigator has enriched my experience throughout my junior faculty years. I have developed long-lasting collaborations and friendship with other scholars through the Beckman Foundation network. I'm grateful for the generous support from the Beckman Foundation, and will always seek the highest level of scientific excellence as Dr. Beckman demands.